Hello students, welcome to today's lesson. In today's lesson, we'll discuss on grade 11 biology about enzyme regulation. The main objectives are explain enzyme regulation and also distinguish activator and inhibitor enzymes. For starting our lesson, let us recap some points about enzyme. Students, what is the role of an enzyme in biochemical reaction? I will give you some time to discuss on it. Let us begin. Well done, students. I hope you wrote some points about enzyme and their action. In the previous lesson, we discussed about proteins. Proteins have different structures. This structure includes primary, secondary, tertiary, and quaternary. Today, we will learn about enzyme regulation. Enzymes are protein molecules that act as biological catalysts. Biological catalyst or biocatalyst means it accelerates the rate of chemical reaction by lowering activation energy. Activation energy is minimum amount of energy required for the reactant to be converted to what? Products. Each enzyme is specific, which means they have a specific active site shape that recognize specific substrate molecules, and they carry out specific chemical reactions. This picture represents the mechanism of enzyme action. As you have seen from the picture, there is an enzyme, and also there is an active site and substrate. So these are the enzyme. This enzyme contains an active site. There is substrate. This substrate entering what? Entering the active site of what? An enzyme. They become bind on the active site. So this active site is responsible to help the substrate to what? To bind. When the substrate binds on this active site, what will happen? Enzyme substrate complex are formed. So the substrate will be attached on the active site. So enzyme changes shape slightly as substrate what? Binds. When the substrate binds, the enzyme will be what? It becomes changes in shape. As a result, it forms products. This product is known as enzyme product complex. Finally, the product leaves the active sites. In case of enzyme regulation is a control system of enzymatic activities in which enzymes are turned on or off depending on what? Depending on the organism's need. A regulatory enzyme. A regulatory enzyme is the one in which biochemical pathway through which it responds to the presence of a certain other biomolecules and regulates the pathway what? The pathway activity. It requires an extra activation process to pass through some modifications and functions. These regulatory enzymes are two types. We call them allosteric enzyme and covalently modulated enzymes. Let us see one by one. Allosteric enzyme. Allosteric enzymes are enzymes that have additional binding sites for what? For effector molecule. This allosteric enzyme, they have additional sites for what? For effector. That causes what? That causes for a conformational changes leading to changes of catalytic property. So in case of allosteric enzymes, if the enzyme contains an active site, beyond into the active site, those allosteric enzyme also forms an additional binding site. On those allosteric binding sites, the effector molecule will be binds. As a result, a conformational change will be takes place. This leading to a changing on what? Catalytic properties. Allosteric enzymes, they contain two binding sites called active site or catalytic site and allosteric site or regulatory site for binding substrates and effectors respectively. The effectors are binding on the allosteric or regulatory sites, whereas the substrates are bind with in the active or catalytic site. Effectors are small molecules. They act as an inhibitor or activator modulating the enzyme activity and functions through reversible non-covalent binding of a regulatory metabolite in the allosteric site or non-active site. As I have mentioned earlier, in case of allosteric enzyme, beyond the active site, there is also additional binding site. That site is known as allosteric site or regulatory site. This helps for the effector to be what? To be bind. So if this effector binds on those sites, it acts as an inhibitor or activator. Effectors lead to conformational change in a concrete part of the enzyme that affect the overall conformation of the active site, causing modifications. If this effector attached on the enzymes, it mainly changes or modifies, or it forms a conformational change on those enzymes, so that these effectors lead to conformational changes in a concrete part of the enzyme that affects the overall conformation of the active site and causing modification in the activity of the reaction. For example, phosphofructokinase. This phosphofructokinase is regulatory enzyme in a process known as glycolysis. So phosphofructokinase, it converts fructose 6 phosphate to fructose 1,6 bisphosphate. So phosphofructokinase is a regulatory enzyme in glycolysis. It helps to convert fructose 6 phosphate to fructose 1,6 bisphosphate. In case of allosteric inhibitor, which means if there is the production of ATP is high amount of energy are produced. As a result, these allosteric inhibitors or effectors, they decrease the products of phosphofructokinase. In case of allosteric inhibitor, this phosphofructokinase, a regulatory enzyme which helps in glycolysis, it converts fructose 6 phosphate to fructose 1,6 bisphosphate. In case of allosteric inhibitor, which means if that process produces an energy, if there is a high energy here, ATP, as a result, they decreases the products of phosphofructokinase. As a result, what will happen? It lowers the process of glycolysis. It is an inhibitor. So allosteric inhibitor, in case of phosphofructokinase, they have done what? They decrease the products of phosphofructokinase. As a result, this decreasing of phosphofructokinase, it lowers the process of what? Glycolysis. In the reverse, if there is allosteric activator, which means if the amount of energy becomes low, or if the energy is as in the form of AMP, adenosine monophosphate or adenosine diphosphate, as a result, this allosteric activator, it increases the PFK, which means phosphofructokinase. As a result, it fasts the glycolysis process. So in simple ways, in case of phosphofructokinase, it is an example or applicable example for what? In allosteric enzymes or the works of effectors. As I have mentioned earlier, those effectors are as an activator or inhibitor. and they lead into a conformational changes. So in case of phosphofructokinase, it helps in glycolysis process. These effectors act as an activator or inhibitor. In case of phosphofructokinase, it is a regulatory enzyme in glycolysis. It converts fructose 6-phosphate to what? Fructose 1,6-bisphosphate. In this manner, this allosteric enzyme, as I have mentioned earlier, this allosteric enzyme, they act as an inhibitor or activator. In case of this process, in glycolysis process, if the energy product gets higher and higher, the allosteric inhibitor starts their works. As a result, they decrease the products of phosphofructokinase. In this case, the glycolysis process will become lower. When the energy production, in case of the glycolysis process, will become lower, the allosteric activator start to their works, which means if there is AMP, adenosine monophosphate, adenosine diphosphate, if there is a lower energy in this process, as a result, this allosteric activator, they activate the increased production of what? Phosphofructokinase. This leads into what? The glycolysis process to become fast. Allosteric enzyme, it has two sites, active site or catalytic site, and also allosteric site or regulatory site. On the allosteric site or regulatory site, allosteric effector will bind. In case of the active site or catalytic site, the substrate will bind. Covalently modulated enzymes. Covalently modulated enzymes are enzymes catalyzed alterations of synthesized protein by the addition or removal of chemical groups. Enzyme activity is regulated by removal of chemical group through a covalent bond. That is why it is known as covalently modulated enzymes. Enzyme activity is regulated by removal of chemical group through a covalent bond. Modifications can target a single type of amino acids. This modification in case of covalently modification enzymes will be done. The modification can target on a single type of amino acid or multiple amino acids and will change the chemical properties of the site. Examples of covalent modification. In case of covalent modification, as an example, phosphorylation. Phosphorylation means the addition of phosphate groups to proteins by kinases and removal of those phosphate by phosphatases. This picture represents enzyme phosphorylation. If there is a protein molecule here, if there is what? A protein kinase. So if there is protein kinase, there is what? Addition of phosphate. Whereas if there is protein phosphatase, this protein phosphatase helps for the removal of what? The removal of phosphate group. In case of phosphorylation, it is the addition of what? Phosphate within the presence of kinase enzyme, or it is the process of removing that phosphate group within the presence of phosphatase. This acts in case of enzyme phosphorylation. When you come to example 2, this example represents the regulation of glycogen synthase in diabetes. Glycogen synthase is an enzyme or covalently modulated enzyme that helps to store glucose as in the form of glycogen. Students, as you have learned in the previous lessons, glucose in case of human body is converted into glycogen and they stored in the liver cell. So this covalently modulated enzyme, we call it glycogen synthase, helps to store this glucose as a glycogen on the liver cell. If a person become a diabetic person, this enzyme, it is inactivated by a phosphorylation process, through which this insulin signaling pathway becomes defected, which means no longer production of what? No longer production of glycogen synthase. As a result, a person leads into a disease known as diabetes. Enzyme inhibition. Enzyme inhibitors are molecules that bind to an enzyme and they block its activity. So inhibit means it blocks what? The activity. There are two types of inhibitors, reversible inhibitors and irreversible inhibitors. Let us see one by one. Reversible inhibitor. Reversible inhibitor, it inactivates an enzyme through non-covalent easily reversed interaction. Or in simple ways, reversible inhibitor means activity without permanently altering the enzyme, which means in case of reversible inhibitor, it is not permanent, which means the activity inhibit temporarily, not permanently. Reversible inhibitor can be classified into competitive, uncompetitive, and non-competitive. Competitive inhibitor is, as the word indicates that, it's the molecule that blocks the binding of the substrate to the active sites. These active sites are the binding site for what? A substrate. So in case of inhibition, it's a molecule blocks the binding sites of a substrate to the active site. Or competitive, it competes directly with the substrate for binding to active site. They compete directly with the substrate. For example, if there is a drug known as methotrexate, it blocks the enzyme dihydrofolate reductase. Methotrexate blocks the enzyme dihydrofolate reductase and it prevents the cancer cell what? Growth by interfering with DNA synthesis. In case of cancer prevention, this methotrexate plays an important role by blocking the active sites. Uncompetitive inhibition are binds only to the enzyme substrate complex, but not the free enzyme. It occurs in the reactions with two or more substrate or products. In addition to that, the slow enzyme reaction by binding the substrates to each other. Non-competitive inhibitor. In case of non-competitive inhibitor, it binds to the enzyme already bound to the substrate. In case of non-competitive inhibitor, it decreases the efficacy or efficiency of the enzyme. They do not compete with the substrate for the active site. As an example, cyanide. In case of cyanide chemical, it binds with what? Iron. This iron is located on the heme group of enzymes. As a result, if the cyanide binds with iron, it prevents the enzyme from functioning and causes for the halting of ATP production. Which means that if the cyanide ion gets into our body, specifically on the red blood cell, it binds with what? Iron. On the iron part, there is a heme group enzyme. This group of enzyme, they help us for the transport of what? Oxygen. So on the heme group, if there is a transport of what? Oxygen. As a result, this cyanide ion binds with the iron. It prevents the enzyme from functioning. This cyanide reduces the movements of what? Oxygen. As a result, without oxygen, there is no cellular respiration will be takes place. So it is the causes for the decreasing or halting of what? ATP production. Finally, this leads to death. So cyanide, it is the best example for non-competitive inhibitor. So this picture represents about reversible enzyme inhibition. As I have mentioned earlier, they are classified into competitive, non-competitive, and uncompetitive. In case of competitive, there are substrate and inhibitor. There is also enzyme. This is an enzyme, this is a substrate, this is an inhibitor. So in case of competitive, on the active side, they compete, so the inhibitor will be what? Bind. That kind of inhibitor is said to be competitive inhibitor. So it competes for what? For the active site. There is an enzyme plus substrate, they form enzyme substrate complex, and there is also what? An inhibitor, finally they form enzyme inhibitor binding. In case of non-competitive, there is no a competition on the active site. The substrate will bind on the active site. This is an enzyme. Different binding sites are located here. There is an inhibitor. So in case of this, there is enzyme and inhibitor plus what? Plus substrate. So these three together, they form enzyme inhibitor substrate, which means there is an active site. On the active site, the substrate will bind. On the different binding site, there is also the inhibitor will be bind. In case of uncompetitive, there is a substrate. There is an inhibitor, but this inhibitor is not fitted for this enzyme. As you have seen from the picture, they are not fitted. But in case of here, if there is alterations of binding sites, the substrate will be what? Bind here, and the inhibitor binds here. Therefore, the enzyme inhibitor substrate. If there is an enzyme, if there is a substrate, they form enzyme substrates. Finally, the inhibitor will bind with the enzyme. The enzyme, they form enzyme inhibitor substrate. So, these are the reversible enzyme inhibition: competitive, non-competitive, and uncompetitive. Irreversible inhibitors. Irreversible inhibitors is a substance permanently blocks the action of an enzyme. In case of reversible, it is not permanently, it is temporarily blocks the activity of the enzyme. In case of irreversible inhibitors, it is a substance that permanently blocks the action of an enzyme. That cannot be what? Reversed by removing the inhibitor or adding more substrate. They permanently block the action of an enzyme. Lead to complete loss of enzyme activity. This irreversible inhibitor, they lead to complete loss of enzyme activity. For example, penicillin. Penicillin is an antibiotic that can kill bacterial cell. So antibiotic penicillin irreversibly binds to transpeptidase, which helps for the blocking the bacterial cell or synthesis. As a result, if there is no bacterial cell production or synthesis, the bacterial cell will die. Assessment. Choose the correct answer from the given alternatives. What is the primary characteristics of allosteric enzymes? A. They have a single active site. B. They are regulated by molecules that bind to sites other than the active site. C. They only catalyze reactions in the presence of high substrate concentrations. D. They do not undergo conformational changes. Question number 2. Which type of inhibition occurs when an inhibitor competes with the substrate for binding to the active site of an enzyme? A. Non-competitive inhibition. B. Uncompetitive inhibition. C. Competitive inhibition. D. Mixed inhibition. Question number 3. What are effectors in the context of enzyme regulation? A. Molecules that bind to the active site of an enzyme. B. Products that inhibit enzyme function. C. Substrates that increase the reaction rate. D. Molecules that alter enzyme activity by binding to allosteric sites. Students, I will give you 3 minutes to discuss on this question. Let's begin. Well done, students. I hope you attempt all the questions. Let's do it together. Question number 1. What is the primary characteristics of allosteric enzymes? A. They have a single active site. B. They are regulated by molecules that bind to sites other than the active site. C. They only catalyze reactions in the presence of high substrate concentration. D. They do not undergo conformational changes. The answer will be B. In case of primary characteristics of allosteric enzymes, they are regulated by molecules that bind to sites other than the active sites. Question number 2. Which type of inhibition occurs when an inhibitor competes with the substrate for binding to the active site of an enzyme? A. Non-competitive inhibition. B. Uncompetitive inhibition. C. Competitive inhibition. D. Mixed inhibition. The answer will be C. A type of inhibition when inhibitor competes with a substrate for binding sites or binding of an active site of enzyme is a competitive inhibition. Question number 3. What are effectors in the context of enzyme regulation? A. Molecules that bind to the active site of an enzyme. B. Products that inhibit enzyme function. C. Substrates that increase the reaction rate. D. Molecules that alter enzyme activity by binding to allosteric sites. The answer will be D. In case of effectors, the molecules that alter enzyme activity by binding to the allosteric sites. As a summary, enzyme regulation is a complex process that involves various mechanisms to control the rate of chemical reactions. Molecules that increase the activity of enzymes are activators, and molecules that decrease the activity of enzymes are inhibitors. Competitive inhibitors bind to the active site of the enzyme competing with the substrate. Uncompetitive inhibitor binds only to the enzyme substrate complex. Non-competitive inhibitor binds to an allosteric site. Irreversible inhibitor cannot be reversed. Students, this is all about today's lesson. Until next time, goodbye.